Thank you for the introduction and opportunity to present today. I'll tell you about the project from Boston Children's Hospital, how we've been utilizing computation infrastructure in support of emerging biomedical science at our institution. My name is Piotr Sliz. I'm the Chief Research Information Officer at Boston Children's. I'm also an Associate Professor of Biological Chemistry and Molecular Pharmacology and of Pediatrics at Harvard Medical School. Before we get started, quick disclosure, we've been a customer of NVIDIA as well as VAST for many years. And in particular, last year, we've benefited from the VAST loaner program to obtain storage in support of our cryo-electron microscopy project on the spike coronavirus protein. And I'll tell you about it later today in the session. If you have any questions or follow-up, please feel free to reach out to me by email or on the social media. I will be happy to follow up and connect with you eventually in person as well. And without further ado, let's get started. So I wanted to provide a very brief background about our research environment at Boston Children's Hospital. And we've been privileged to be ranked #1 hospital in the nation for 8 years in a row, and also awarded the Healthcare Equality Index leadership status for 5 years in a row. So in particular, very proud of those two achievements. For those of you who are not familiar with the Boston area, we are located in Longwood Medical Campus, right on Longwood Avenue, in a close proximity to many local hospitals, and in particular Harvard Medical School. Boston Children's has a regional network and satellite locations as well, so our regional presence is really strong. But beyond that, if you look at this next slide, we've been also involved in many global consortia projects and operations and spanning virtually from every single country globally. So I wanted to kind of mention from the innovation and collaboration perspective, we have a particularly strong research program, and one of our key drivers here is genomics research, rare diseases in general, world-class informatics. And in general, we very well positioned to take on this transition from the clinical to translational within the hospital itself. So in this next slide, I just wanted to kind of also show we the teaching hospital of Harvard Medical School, world's largest research enterprise for pediatrics. We have many distinguished members of the faculty, and one highlight here, we just had 3 new members elected to the Academy of Medicine just back in October. So this next slide, I wanted to dive into the first project that I'll describe today is the Children's Rare Disease Cohorts Initiative. And it's just one of many initiatives at Boston Children's Hospital. And just like any other one, it benefits from many research areas and many research programs that we have at the institution. One of the biggest drivers, I don't know if the biggest driver, but one of the really key motivation for the CRDC initiative was this project by Tim Ulap. And that project, you can see a reference to the paper that was ultimately published in the New England Journal of Medicine. And this was one of the first examples when we've really taken the approach to develop this personalized medicine for a single patient and do it very efficiently within 10 months in this case. This is 10 months from the idea to injection. Definitely the drug, the treatment made the difference for that particular patient when it was used. And at about the time when Tim was working to develop that drug in 2019, David Williams, who was at the time the Chief Scientific Officer of Boston Children's, also started the effort through this Blue Ribbon Committee to really kind of come up with the ideas how to do genomics effectively and efficiently in our community. And one of the main outcomes from that was to really focus on the rare diseases. And this ultimately led to our research strategic plan in 2019, and the key recommendations were to optimize the patient consenting to ensure that the data, when it's collected, can be more broadly utilized across different diseases, can also be collected in a CLIA fashion, which can support clinical decision-making if needed, and the data is kind of more broadly harmonized and processed in the same way across the hospital to ensure that everybody can benefit from the database. So that ultimately led to the CRDC project that started in 2020, and now it's in the second year, and I'll give you updates about where we are with it today. And just kind of from the synergy with what we do at the Children's, this was extremely good fit because a lot of our clinicians were already involved in rare diseases. A lot of patients come to us, and you can see here those numbers, sometimes for 5 years without having proper diagnosis and coming to us. And then with the help of our clinicians, with the help in many cases of genetic interpretations, we can help them to converge with those diagnostic odysseys that they've been undergoing for many years. So couple more patients here to highlight. Matthew Elliott, who's been treated at Boston Children's in 2013 for ALD, and more recent case from 2019, hemophilia A patient who've been also successfully treated with a therapy clinic at Boston Children's. Many of you have probably heard of the children's rare disease approach, which is in general very similar to precision medicine approach, which effectively involves looking at your genome, one variation, one gene at a time, trying to combine it with the phenotypic information, environment, lifestyle, etc., and come up really with personalized medical approaches. And we really take it one step ahead of that at Boston Children's. And here, by virtue of having our very strong biomedical community, we can actually take the variation in DNA and then explain how those variations really translate to the protein variations. And proteins are the smallest functional element of your body. Changes dynamics of the proteins. They can act as catalysts. They can act as scaffolds. Changes in those proteins are driven by the variations in your DNA. So really truly understanding how those changes affecting your body, affecting your function, help us to then postulate the therapeutics, therapeutic approaches and treatment. I mean, that's the holy grail of the whole approach. And we've been lucky to have this both genomic community as well as mechanistic community at Boston Children's. So later in the talk, I'll come back to some examples of how the structure determination can be really driven to support science. And here maybe most importantly, so this is kind of from the functional approach, I wanted to mention that children's rare disease cohorts very broadly, and that's probably the reason for me to be in this session today, very broadly depends on the computational approaches. And being scientific software, in our case provided by BioGrids, being expertise of our data science team, being computing resources, storage resources, all of that has to be put together to allow us effectively and quickly analyze the data sets and drive the conclusions to support clinical and research for our community. So I wanted to kind of now break into three different components. We're looking from the children's rare disease cohorts, components that are really foundational to our effort, and they include data, digital infrastructure, and analyses. And in particular, data acquisition and data, it's critical. And it's critical for genomics, it's critical for structural biology and many other fields. And as you can see on this next slide, the community that contributes to the data acquisition and effective data collection is really enormous. We have a group of over 30 clinicians right now working directly with patients, making sure that we identify rare and interesting cases that can benefit from the genomic analysis, but also explaining to patients how this data will benefit potentially their treatment and research in general. It's really fundamental here. We have the infrastructure team that is helping us to really work with those large amounts of data. And here we involve people who are worried about the data acquisition, data processing, data security, making the data shareable in the local Boston environment to support science that is needed. And on the left-hand side, data science team that has been really working with us and driving the project, in particular, Fearless Shira Rakovitz on the very left to make all of this click. So this next slide, I have this research to clinical pipeline, and that's kind of to reiterate complexities of the data collection. On the very left, from the consenting, we have to ensure that when data is collected from our patients, that is consented by patients for the particular use, and it this consent really allows us to more broadly analyze the data in context of the particular disease, but also in support potentially of other research projects. The blood collection or saliva collection has to happen effectively, and this has been driven by ability for us to deploy last year red cup systems to do at-home consenting by patients, but also the collection is done from the home environment. So patients, especially last year, were not very keen to come to the hospital. We were able to sustain it and continue the project. And then the data with the proper identifiers go to the sequencing facility. We can sequence it very quickly, bring it back to our research databases if we have clinical findings to then reflect it again and get those clinical confirmations from the sequencing vendor to assure basically quality of data for the purposes of patient treatment. So this next slide, just very quickly, the 2020 status report, just to show you some numbers. So we've collected over 1,600 exomes supporting 1,600 patients with different rare diseases at BCH. In addition to that, we've collected over additional 4,000 samples from their families to support analyses. You can appreciate from that 16 groups, research groups involved with the analyses, over 75% of the data analyzed by the end of the fiscal year. So very aggressive timeline. And in addition to the clinical follow-ups, decision-making, many publications and grants right now already published or in preparation resulting from the research projects. So that's from the kind of a data perspective. From the data digital infrastructure perspective, very quick alignment, again, of the hardware we have, software, and the needs of the community. So I wanted to kind of quickly go here over 5 challenges as I see it from the project. The challenge number 1 is data harmonization, and this is broader than genomics. For any other field, the challenges are similar. But in particular in genomics, we need to be able to reanalyze this data on a monthly or at least twice a year basis to take advantage of the new reference data sets, to take advantage of the new software approaches. And to do it effectively, if we do it on the commodity hardware, it would take us over a year to do it with just the collection we have today. So we've been working with Illumina, for example, to develop this, to deploy this FPGA array hardware internally when we can accelerate this process and be able to process data sets within a month or so. We also in parallel working with NVIDIA Parabricks to be able to accomplish the same and comparing the outcome. Another challenge is data integration, combining the genomic data sets with other sources of information, electronic health records, clinical notes. We've deployed RedCap in support of electronic data collection. Cliniting, it's supporting natural language processing, NLP from clinical notes. And we've deployed CoreDB, WishNextCode technology for very effective and fast relational database that allows us to access all this information for analyses from various systems. So challenge number 3, all of that, analysis, downstream analysis, once we have the data integrated and the same bucket, has to be done actually very quickly as well. In particular to support evolving research and patients follow-up. We want to, in general, be able to do analysis within weeks from the time when data becomes available. So we've deployed a number of tools. We have WishNextCode, and this has been our kind of tool number 1 from day 1. More recently, we developed Imagine, which supports machine learning prioritization of variants and is really helping clinicians to do most of the analyses as far as we can process automatically. We also working with the Broad Institute to deploy Seeker. We have currently an instance of Seeker, but it's being extended. So as you can appreciate from that, the challenges from the data perspective is to ensure that all those resources are connected to data sources and being on-prem or in the cloud environment. It has to be efficient, it has to be secure, and it has to provide results quickly. So there are additional layers of complexity, and challenge 4 here is really working with diverse and evolving analysis workflows. So as I mentioned, we have a couple of the standard software applications which kind of take care of 90% of what we do. The remaining 10% is addressed by this large collection of BioGrid applications, over 400 of them right now. And this supports projects when people bring additional data types or do deeper, more customized analysis. And this is done on the computational resources within Boston Children's in our local data center, which are our regional MGH PCC center, which is utilizing also NVIDIA and vast computational hardware. And most of this effort led by Arash Khayati. And here to kind of stimulate you to show you where this is heading in the future. So from this analysis, when we look at one genome at the time from a single patient, moving forward, we're trying to really break it down and try to do it on the cell level. When we look really at the specific tissue or disease environments and try to do the cell-based analyses. Enormous projects, and we just got funded for one of those by Chan Zuckerberg Initiative Award, just announced in September, with Jay Tyagarajan leading that effort, and I'm involved in the project as well. And we currently trying to deploy one of the CZI technologies, cell by gene software, to help that local analyses to help us build the infrastructure to be able to analyze those larger data sets. Just kind of across the board, I hope that just going over those 5 challenges demonstrate complexity of our operation and also necessity to be really at par with the current requirements and emerging techniques in the field, which happen on an almost daily basis. So just kind of coming back to this slide to show you how this data analysis integrate with resources and access to our vast databases, genomic databases. I wanted to dive very quickly to highlight one of the projects that was done by our clinician, Dr. Janet Chow. And Dr. Chow was, before COVID in 2019, already started to work with us. She was collecting this very focused but small collection for patients in the cohort around severe immune disorders. And by 2020, she had 48 patients in this cohort with fairly high diagnostic yield of 53%. So when COVID started, together with other investigators, she started to also focus in particular on sequencing of patients with more severe cases of COVID, of MIS-C cases, which started to really trickle at Boston Children's Hospital. And here on this graph, you have analysis from the TriNetX network, which integrates healthcare data from across US. And this was done in 2020. And as you can see from across the adult population, those pediatric cases were much more rare in general, and in particular severe cases were really at the time very limited. And information how to treat it at the time when the COVID started was really almost not existent. So in this study highlight, I wanted to mention that as part of this analysis, and we just happened to have the sequencing from one of the patients who was previously in the severe immune disorders cohort, the patient was shown to have the SOX1 mutation. And as you can appreciate from the picture of the structure of that complex proteins, which includes SOX1, the SOX1 is here colored in red. Effectively, mutation to that protein would disable formation of this larger complex, which is typically required in healthy individual to support interferon pathway. So without that protein in place, this was basically equivalent of having inhibition of JAK kinase. So with this molecular understanding of the pathway, Dr. Janet Chow was able to deduce the mechanism of that severe disease in a particular patient, adjusted treatment, and successfully treat the patient. And that work also resulted in the publication led by Janet Chow, which was published just recently in Journal of Allergy and Clinical Immunology. So this is just one of the highlights. On this slide here, I wanted to demonstrate that we have many examples, like the one from Dr. Janet Chow laboratory, and in particular, we have many really emerging collaborations where we can see clinicians from different patient cohorts working together, making new discoveries or coming up with the treatment for more complex or overlapping diseases. So I wanted to switch gears a little bit here right now. So I've mentioned from the CRDC initiative at Children's, we've mostly focused on the genomic analysis. But as I've mentioned earlier, the variations of your genome is really informing proteins, which are eventually the smallest functional blocks of your body, how those proteins are affected by the genomic variations. And the tool that has been utilized by the community to support that analysis is structural biology. So at Children's, we really have a long history of structural biology, which was initiated by Professors Stephen Harrison and late Don Wiley back in 1970s. And by now, we have many other investigators who are involved in structural biology research, including Beng Chen, Sun He, Tim Springer, and Hao Wu. And this is truly important for our computational analysis because we've been really blessed with access to, in many cases, most challenging and vastly interesting structural biology projects and structural biology structure determination efforts in the community. And this extends beyond children's to our local community at Harvard Medical School as well. But having access to the data from those research groups really allows us to fully tune up and improve our computational infrastructure. But what I wanted to highlight on this slide is the infrastructure team, which is really with my involvement led by Jason Key and Pete Myers and others. This infrastructure team, we call it SBGrid, Structural Biology Grid Consortium, was initially developed with the aim to supporting local effort. And over the years, it expanded now dramatically to support the global community with over 400 research groups participating or taking advantage of this infrastructure, software infrastructure developed by Jason and others. This next slide here, I wanted to highlight a couple of projects that I've had the privilege to be involved with back in 2004, 2005. And this included structure determination of cluttering codes, aquaporin 0 complex, and having access to the software infrastructure at the time developed by SBGrid was really critical, but it also provided for SBGrid opportunity working on those at the time cutting-edge, high-resolution cryo-EM models allowed us to really fully develop our collection of software in support of cryo-EM efforts. So fast forward to 2021 right now, SBGrid Consortium, it's present in over 22 countries. We have a collection of over 450 applications in the collection. And still the major effort, the major focus of the consortium is to really establish the scientific software portal and scientific software store for the structural biology community. So here on the left, you have a quick view of our SBGrid software installers. Any of the labs who participate in the consortium can have access to that tool and very quickly download any of the 450 or so applications that we provide. Applications come precompiled and ready to execute, so scientists don't have to waste time working, tweaking up their environment. They can take advantage of those tools immediately. And they also, on a daily basis, have access to the tools which are released by our developer community. In parallel, we've also developed very extensive training materials on our SBGrid YouTube portal. We have currently over 200 training videos which have been developed with participation of software developers from the community. So this next slide here, I just wanted to show you the complexity of software, of our software in the cryo-EM field. We currently have over 100 applications specifically supporting cryo-EM, and you can appreciate they support, in many cases, different phases of structure determination. Some of the packages are more comprehensive, trying to more globally help people streamline processing. But with this rich collection, changes to the applications, updates are presented to us on a daily basis, and we make sure that those software tools are propagated to the community. An additional challenge here is also a lot of the software application, in particular in the cryo-EM, are taking advantage of the hardware acceleration, GPUs, and evolving hardware technologies. And part of our job is to ensure that we optimize those applications to tweak up and provide the best performance for our community, to allow them to focus on the science. So in this slide, I wanted to kind of reiterate that connection between the computing and data acquisition at Harvard Medical School, where we established a very large cryo-EM data collection facility with the big microscope here picture in the center of that slide. This kind of extends from our PIs, and here Bing Chen on the left preparing his sample for the cryo-EM collection. On the right-hand side, I wanted to kind of highlight our computational infrastructure that was developed by Sean Rawson and other members of SBGrid to streamline processing of the cryo-EM data sets. And what was critical here is this approach to on-the-fly processing. A lot of our computational resources are deployed within 30 feet of the cryo-EM data collection instrument in the adjacent room, and we can very quickly stream all the data. And as the data is being collected, the analysis starts typically within 30 minutes. And within the 2 hours of initiating data collection, in many cases, we can already process the data sets and start to get a sense of the quality of data that is being required. And that's important because the data collection time is extremely expensive. Sample preparation time takes a lot of effort. So with this very quick feedback right at the data collection facility, we can adjust our collection strategy and optimize it for maximum effectiveness. So I wanted to mention here, we have a fast storage deployed in support of that. And beyond the cryo-EM facility hardware that we have deployed right on the edge of the facility, we also take advantage of other regional, institutional cloud resources to do further processing. But balancing between this in-house and external processing resources remains the challenge. So now here, just kind of a highlight of one of the projects, and this is from Bing Chen's lab, and that's a project I've mentioned in the openings that benefited from the vast loaner system. And Bing was already working in this area, so it was kind of a quick extensions to him to try to answer some of the key questions around COVID, and in particular here, he studied this D614G to glycine variant, also known to you as Delta variant. And he determined the structure of the variant by cryo-electron microscopy. And this finding was, the results were published in Science just earlier this year. And he explains here how this transition from the pre-fusion state on the left-hand side, when you can see the virus with the spikes on it, and in the fusion state, it's merging now to the membrane, and small variations in a spike protein lead to the disorder in the loop region, and then the details of that you can see here on the slide on the right. I don't provide all the details here. You can go back to the paper and read about it. But long story short, the variation and change to the disordered loop changes basically affinity of the spike protein to be able to fuse with the human membranes for the entry of the viruses into the cell. And with the Delta variant, it ironically just becomes much more effective in this fusion process. So that fundamental study by Bing Chen really helped us to explain that mechanism, and therefore now can lead to future improvements and development of therapeutics. So that's one of the projects that get done at the facility. By now we have a portfolio of approximately 10 really high-profile structures that emerged from this facility over the last year. And our aim is to remain being very efficient and effective and help scientists to make a quick analysis actually happen and help to support healthcare globally with the resources we have on hand. So just with the summary here, so I wanted to kind of reiterate, go back with the high quality on scientific data sets is the key for us, and privileged to work with the local community to have access to those excellent cryo-electron microscopy data sets. I didn't talk today about the lattice microscopy. That's another field that we are also very deeply involved with, or genomic data. And having meaningful data sets to really help with research and clinical aspects of what Children's Hospital delivered has been really important and a driving factor for our computational efforts. So in terms of digital infrastructure, I've talked about the different aspects of that. Most importantly, I think the scientific software, which is developed by the global community of developers, is the key to making quick progress and advancing research in many different directions. And SBGrid and BioGrid's effort from Children's and Harvard Medical School provide an umbrella to be able to have that collections of the curated, ready-to-run applications that can be used by any scientist in Boston area as well as globally. Edge resources, this on-the-fly processing that I've highlighted, it really supports our cryo-EM analyses. We've used this similar approach to some degree with genomic data sets and lattice microscopy, other approaches, and building those customized computational infrastructures with really fast software, single use case scenario initially is critical to really have the environment that supports the field to move forward as quickly as possible. In phase 2, typically we try to generalize it and then go to the more global local resources which support general computing. But having this special infrastructure has been essentially a key for us to make quick progress in the fields that we support. And we obviously also very strongly engage with the remote resources, and this might include downstream processing of data management. We collaborate with Harvard Medical School to have the tiered storage for cryo-EM data sets. We also work with the regional MGH-PCC effort in collaboration with Harvard University, BU, and we have presence in AWS, Google, and other cloud services. And last but not least, the domain expertise, again, the privilege of working with our local investigators, essentially is the key here. Having been a member of the consortia like SBGrid, it really helped us to come up with the best practices for the community, reutilize shared infrastructure, and support many projects effectively, and having in combination training and educational programs have been enabling for us and for the community. Thank you for your time and attention today. I wanted one more time to acknowledge everybody that contributed in many different ways to the project. In particular, Shira Rakovets, Dr. Janet Chow, and Beng Chen, who led those projects. I'll also highlight it again. So please feel free to get in touch with me by email, as I mentioned in the opening, on social media. Happy to connect with you now and in the future. And thank you one more time for your time and attention.